And I'm going to introduce Teresa Lambe, who's, as I said earlier, an Associate Professor and Jenner Investigator from the Jenner Institute, Oxford.
Thank you very much, Sheila, and thank you very much for the organisers for inviting me here. I'm, as introduced, Teresa Lambe, or Tess. I'm from the University of Oxford. And Sheila's absolutely right in that we've been working and developing vaccines I've been in the field for 12, 13 years now. And one of the vaccines that, vaccine technologies that I mainly focus on are adenoviral vaccines, and we refer to them as platform technologies. So what do I mean by platform technology? Well, it's a technology that we can rapidly adapt to make a vaccine against any emerging or outbreak pathogen. So how do we do that, and why do we use adenoviruses? So as Sheila quite nicely summarized, thank you, adenoviruses are a great platform technology for inducing a really strong immune response, so inducing strong antibodies and T cells. And antibodies can be thought of the part of the immune response that will clamp onto the virus, the invading pathogen, and stop it getting into your cells. So they're neutralizing antibodies. And then T cells are another part of your infiltrate that will prevent systemic infection. So T cells will come along, will recognize infected cells, and will attack them, will kill them. So you don't get a lot of virus being released into your body, and you don't get overtly sick. So they're the types of immune response that we want to induce. So how do we do it with our adenovirus? So we work with a chimp-derived adenovirus, and the reason that we use a chimp-derived adenovirus is to circumvent the pre-existing immunity that we may have at a population level. So as Sheila had rightly pointed out, that everybody in this audience has likely been exposed to an adenovirus before because they cause a common cold, an upper respiratory infection. And that can mean that if you use it to make a vaccine, a common adenovirus from a human, it can result in a dampened immune response. So what we've done is we've adapted an adenovirus from a chimp, and we've modified it so it doesn't replicate. You can't get a common cold from this adenovirus. We've crippled it, we've knocked the legs off it. So when a disease comes along, or an emerging or outbreak pathogen comes along, and we want to make a vaccine, we have our platform technology, it's ready to go. We know it can induce strong antibodies in T cells, but what we don't know is what part of the pathogen we should use to induce a strong immune response. We typically will look at the pathogen, look at the literature, have lots of conversations over cups of tea, and decide what part of that pathogen we think will induce the right type of immune response. So neutralizing antibodies, strong T cells that will protect us against overt disease. In the case of SARS-CoV-2, the antigen, or the small part of the adenovirus that we chose, was the spike protein. And that's the part of the SARS-CoV-2 virus that's on the outside of the virus that mediates its entry into a cell. So we took the coding sequence for the SARS-CoV-2 spike protein, we stuck it into our adenovirus, into our platform technology. And that means that when we get injected with that vaccine, we will mount a strong immune response to this small part of SARS-CoV-2 that will be expressed in your body, and we'll get strong antibodies and T cells. So how did this all happen so rapidly? Because you'll have heard that normally it takes about 10 years to develop a vaccine. Well, it starts from predominantly me being an overprotective big sister. My brother was based in Shanghai at the time of the SARS-CoV-2 news breaking. I was fastidiously following any kind of outbreak pathogen in China because I work in the field of vaccine development, and I'm a protective older sister. So once the genetic information came, the recipe to make the last missing piece that we needed to make our vaccine, that came through over email on the 10th to 11th of January. Myself and my colleague designed the vaccine over the weekend, and we ordered the reagents we needed to make the vaccine that Monday, and we were ready to go. And we were ready to go at pace because we had invested all this time in this platform technology. And we were able to start our preclinical studies, so the studies that you need to do in animals, really quickly. And the reason we were able to do that is because we had a huge network of collaborators. And that's probably the tale of what we've learned through this pandemic. It's through concerted effort and collaboration, with everybody coming together with a unifying drive to try and beat this virus, that we've been able to move so fast and so quickly. So we did all the normal steps that you would do during a clinical trial. And the only risk we took was a financial risk, not me personally, but the financial risk from the UK government, investment from AstraZeneca. Serum Institute, one of the largest vaccine manufacturers in the world, also took a risk that our vaccine would be effective or efficacious. We did not know that at the time when we started our small-scale phase 1 clinical trials. We obviously do know it now with real-world effectiveness data coming out showing that the AstraZeneca and the Pfizer-BioNTech are as effective in the real world, and we're able to protect our most elderly and our most frail from overt disease, hospitalization, and death. So what did I do during the phase 1, phase 2, and phase 3? Well, I did an awful lot of antibody testing, and that's been my life for the last year or so. So I helped develop the antibody tests that we use in Oxford. I worked as part of the team in developing the clinical trials, and I oversaw the immunological readouts that were required to progress this vaccine onto phase 3 and efficacy readouts. So that's a vignette into what my life has looked like in the last year and a half. The last parting comment that I'll leave with is that we could not have got where we are today with so many vaccines being available if we had not worked as a team. So this pandemic has brought out the best in a lot of us. It's thrown up an awful lot of hardship, and it's not over by any means. But when we work together with a common goal and a common purpose, we're able to do some really remarkable things. And that is something that we should not forget going forward. And I'll leave it there.
Thank you, Tess. That was really interesting. I think that's a really important point. Collaboration is the name of the game. I wondered if you might want to make some comment about the initial funding of the project. The audience might be interested in how such a massive project was able to come about in the first place.
Yes, so I've never been a person who's been very secure when it came to funding, because I worked on diseases that probably a lot of the audience haven't heard of. So diseases like Crimean Congo hemorrhagic fever, which is quite a niche field. You probably have heard of Ebola, you may have heard of Lassa, and you'll have heard of Zika. So vaccine development up until a year and a half ago was seen as almost stale. It wasn't seen as exciting, and these emerging and outbreak pathogens were generally thought of as diseases that happened far away. They wouldn't impact us. So getting funding was always quite difficult. And we did a lot of the funding of these types of activities, including generating the data around making this platform technology from grants. So they're either charities or UK government that will fund this type of research. And that's again where we started with the coronavirus vaccine, the ChAdOx1-nCoV-19 AZD1222. Just rolls off my tongue now. And then we obviously knew that we had to bring in partners to be able to do these large scale clinical trials to fund the GMP manufacture. So for example, going from small scale animal studies to first in human, that takes about £1.5 million. So they're not small amounts of money. So we did an awful lot of fundraising in the early days, along with generating the science.
Excellent. Okay, thank you very much. That was